For the past 5 years, a team led by Shizheng Li and Kai Ji at the Wuhan Institute of Virology in China has sampled thousands of horseshoe bats in locations across China. They sought out caves in remote areas looking for strains of the coronavirus that looked similar to human versions. Researchers spent 5 years monitoring the bats that lived there, collecting fresh guano, bat excrement, and taking anal swabs. They were conducting genetic mixing among different viral strains to assess the risk of spillover into populations and emergence of a disease similar to coronavirus. In the last decade, bats have been recognized as an important natural reservoir for zoonotic viruses affecting both humans and livestock. Both RNA and virus families have been detected in bats, including coronaviruses. The quest for a virus discovery is to detect viruses, and also determine transmission pathways, especially to humans. Bats are likely to spread some viruses through feces and urine. These are materials that can be highly infectious to humans. Therefore, the studies focused on collecting freshly voided bat feces for molecular detection of multiple virus families. Also, the presence of viruses in bat feces helped to shed light on the risks of virus transmission to humans, especially through direct hand contact with freshly voided feces and urine. To study the risk of bat coronavirus emergence, serum samples from 1,596 residents enrolled in a study from 2015 to 2017. The serum samples were collected and analyzed. It is unknown how many participants were enrolled in 2018 and 2019. Previously 240 participants were from Wuhan in 2015, and more recently 100 were enrolled from Wuhan. It is like that there were more enrolled since the primary research was being conducted at the Wuhan Institute of Virology in China. Without funding from the National Institute of Allergy and Infectious Diseases, award number ROE 110,964, this study could not have been conducted. Funded came from EcoHealth Alliance Incorporated in New York. The Wuhan Institute of Virology in China has been able to conduct studies with little oversight. EcoHealth Alliance Incorporated received $733,740 from the National Institute of Allergy and Infectious Diseases in 2019, and has received total of $3,741,715 since 2014. The risks are tremendous when conducting studies in a biotech scientific laboratory that is less than 1,000 feet from the Wuhan wet market. During one of the studies 155 bats were captured in Hebei province, and another 450 bats were captured in Zhejiang province. The work from the study was supported by the United States Agency for International Development, the Institutional Review Board Administration of University of California, along with others. The complexity of the study included testing serum samples and diluting each of them with 4 different coronavirus. During the study, microtiter plates were coated with recombinant, and if a bat recombinant protein was used, there would need to be extensive safety measures and protocols in place. Unfortunately, studies often overlook the fact that participants need to complete serologic tests that look for antibodies in the blood of the participants, which is often omitted in order to save money. These serologic tests are used to diagnose various disease conditions and ensure that participants are not infected during the study. It has been determined that the current coronavirus in Wuhan is 96% identical at the whole genome level to a bat coronavirus, so undoubtedly it came from bats. Currently many officials are attempting to debunk that the cellular level biosafety level 4 facility with a high level of operational security, that works on researching dangerous pathogens, has made errors while researching the coronavirus, that has possibly led to grave and deadly consequences. The Chinese have an old proverb, muddy waters make it easy to catch fish, and for now, the world is isolating themselves from China, and unfortunately treating the situation more like an endemic than a pandemic. At this point there are many missing links to the puzzle that have yet to have been revealed.